So to start, I want to put everybody in the mindset of somebody who studies cell biology every day. So cells function like machines, and proteins are the gears and the switches that let these machines work. Now, in a normal cell, there's a protein RAS that acts as a master switch for cell division. So when a cell needs to divide, it first has to turn on this master switch RAS. And now RAS interacts with a second protein, RAF. And when these two proteins are interacting, it means that the cell is dividing. And this is important because cell division is a big part in cancer, and these two proteins are often mutated in cancer cells. So our goal with this project was to determine how these two proteins interacting plays a role in cancer. So what we did was to take each of these two proteins and tag them with a bioluminescent tag. So when you think of bioluminescence, think of what makes a firefly glow. So we were able to take our first protein, RAS, and tag it with a bright yellow glowing tag, and take our second protein, RAF, and tag it with a bright blue glowing tag. Now what really makes this assay so powerful is that this yellow tag will only be glowing if RAS and RAF are interacting. So what this means is every time we see a yellow glow coming from the cell, we know that this cell division machinery has been turned on and that the cell is dividing. And so why is this important and why should you care? And the ultimate goal of this project, again, was to determine how this interaction plays a role in cancer. And this protein, RAS, is mutated in over 30% of all human cancers, and over 90% of pancreatic cancers in particular, which are known to have a very poor prognosis and very few treatment options. And so what we found was that depending on how these proteins interact, it changes how cell division occurs. And so ultimately our goal was to take this information and move towards more nuanced treatments and therapies based on individual mutations in these cells.